Hello everyone, Assalamu alaikum. Welcome to my YouTube channel. This is me Faraz Qurban Rajpur, and in this video lecture I am going to discuss with you about the androgens or testosterone. Androgens. Androgens are basically the male sex hormones, and they are the steroid hormone. Like other steroid hormone, they are being synthesized from the basic precursor, that is the cholesterol, which you know that is the basic precursor for the all steroid hormone, that is the C27 carbon nucleus present in the cholesterol structure, which will undergo a series of different reaction pathway to be converted into the different type of steroid hormone. Androgen inside the human body is being synthesized from the different organs. Like in the male, it is being mainly synthesized from the testes, and beside the testes there are other sites in the human body from where androgen or testosterone are being synthesized, like adrenal cortex, liver, and in the female small amount of androgen is being secreted from the ovaries. The basic carbon nucleus that is present inside the androgen or testosterone is the C21 steroid. They are being synthesized from the C21 steroid nucleus, that is the pregnenolone nucleus. We will observe in the upcoming slides that how pregnenolone is being converted into the testosterone. Classification of androgens. Androgens are mainly classified on the basis of their physiological activity or biological action inside the human body. Like they are androgenic or male characteristic promoting activity, means these are those androgens which have mainly the androgenic activity, means they are basically involved in the development, functioning, and maintenance of male sexual characteristic and the development of secondary sexual characteristics. Anabolic activity. Androgenic hormone are those which have the anabolic or muscle building activity, means they have the synthetic function or anabolic function inside the human body. Like they are mainly involved in the retention of nitrogen, and we know that whenever the retention of nitrogen or nitrogenous compound taking place inside the body, they will ultimately being converted into such type of nitrogenous compound, like protein is classical example. Protein synthesis will occur, and as protein synthesis is occur means protein catabolism is being reduced. So inside the body, these anabolic androgenic hormone is facilitating the protein anabolism and decreasing the rate of protein catabolism, means degradation rate of protein is being decreased, and this will ultimately facilitate or stimulate the thickness and the linear growth of bone to some extent. And besides that, they are also useful in the other conditions of the body. They are like the cancer, trauma, osteoporosis, and the immobilization in these being the treated by the different type of anabolic androgenic steroids. There are different type of commercially available anabolic androgens. The examples are oxymethylone, oxandrolone, and nandrolone decanate. Beside having the anabolic function, these type of commercially available androgens have also some significant androgenic activity, which is related with the secondary sexual characteristic of the males. So, if such type of anabolic steroid are being used in the treatment of certain women patients, this will create the problem of virilization in those patients. The synthetic derivative of such anabolic steroids are being made, like one is made by the 17 alkyl group substitution. Some are the orally active formulation, and though the orally active are those which have the 17 alkyl substitution. And there are certain other synthetic derivatives which are being made without the 17 alkyl functional group, and those are the nandrolone decanate. They are only active intramuscularly. The 17 alkyl group formulation product have the disadvantage of being causing the liver toxicity or the disease or damage to the liver cell if being used for the long time period in the patients. Biosynthesis of testosterone. Testosterone, like other steroid hormone, is basically synthesized from the basic precursor, that is the cholesterol. Cholesterol is the carbon 27 molecule. It gets converted into the series of reaction into the carbon 21 nucleus or the molecule, that is the pregnenolone. Pregnenolone has a carbon side chain at the C17 position, which we have already discussed in our other video lectures. For converting into the testosterone, this pregnenolone carbon side chain, which is present at the C17 position, is being removed. That means pregnenolone, which is basically C21 molecule, is now get converted into the C17 molecule by removal of the side chain that is present at the 17th position. In comparison to the testosterone, pregnenolone has the hydroxyl functional group or alcoholic functional group at the third carbon number, and the carbon-carbon double bond is present in the 5-6 position. But in case of testosterone, we will find the different functional group. Like at the carbon third position, we will find that ketone group will be present. Now let's discuss about the different steps that are involved in the biosynthesis of testosterone. The first step is the synthesis of dehydroepiandrosterone. Dehydroepiandrosterone is basically synthesized from the pregnenolone, which we know that is the C21 molecule. Okay, the pregnenolone will undergo the two different steps. First, it will undergo to be converted into the intermediate product, that is the hydroxylation will take place into the pregnenolone ring nucleus. The hydroxylation will take place at the C17th position, and now pregnenolone will be converted into the 17-hydroxypregnenolone. 17-hydroxypregnenolone is basically the intermediate product. Now this 17-hydroxypregnenolone will undergo through the oxidation. After oxidation, a new product will be formed, that will be the dehydroepiandrosterone. Dehydroepiandrosterone is basically the molecule which have the ketone group at the C17th position, and it also like the C17 carbon side chain that is present in the pregnenolone. So like over here, the reaction is shown in that the pregnenolone is directly converted into the dehydroepiandrosterone. In this reaction, I have not shown the intermediate step, that is the conversion of pregnenolone into the 17-hydroxypregnenolone. Okay, so basically what is happening, this pregnenolone, which you may observe that it has the hydroxy group at the third position, and at C17 position there is the carbon side chain present, which makes it the C21 molecule. Now it will undergo through the two different steps, and then ultimately it will get converted into the dehydroepiandrosterone. You may observe that at C17th position in the dehydroepiandrosterone, which may be abbreviated as the DHAE, there is the presence of ketone functional group, and it does not have the carbon side chain. So dehydroepiandrosterone is basically the C17 molecule, which have the ketone group at the 17th position. Now, in next step, this dehydroepiandrosterone will be converted into the next product. This is the step number 2, which is the formation or synthesis of the androstenedione. Androstenedione is basically synthesized from the dehydroepiandrosterone. Dehydroepiandrosterone, which we have already observed that has the hydroxy group at the carbon number 3rd position, and this carbon number 3rd hydroxy group will undergo through the oxidation, and we know that whenever alcoholic or hydroxy group undergo through the oxidation, it will get converted into the ketone group. So now this dehydroepiandrosterone C3 hydroxy group will be converted into the ketone group, and beside this there is an other interconversion take place inside the chemical structure, which is the tautomerization, which is happening because of the enzyme, which is the 3-beta hydroxysteroid dehydrogenase. This will cause the transfer of this double bond which is present between the carbon number 5 and 6. This bond will be shifted over at this position between the carbon number 4 and 5. This we have already discussed in the biosynthesis of the steroid hormone basic lecture, which I have already uploaded on my channel. So we have already discussed that mechanism in detail, that how this bond is being shifted over at this position. This is basically occurring because of the action of this 3-beta hydroxysteroid dehydrogenase enzyme. So ultimately there are the two changes take place inside the chemical structure of dehydroepiandrosterone. First is the oxidation of this 3-hydroxy group, which get converted into the ketone group, and second is the interconversion of this double bond. And now ultimately this new molecule will be formed, that is the androstenedione. Androstenedione has the ketone group at the C3rd position and double bond between the 4 and 5 carbon. This you may observe. So now the next step is the reduction of androstenedione. So let's recall our memory that first step was the formation of dehydroepiandrosterone from the pregnenolone. In the next step, dehydroepiandrosterone undergo through the oxidation and will get converted into the androstenedione. And now in the step 3rd, reduction of androstenedione will take place. As we have already discussed, the chemical structure of androstenedione has the ketone group at the C17 position. Now this C17 position ketone group will undergo through the reduction, and we know that whenever ketone molecule or ketonic compound undergo through the reduction, this ketone bond will be converted into the hydroxyl functional group. So now this C17 keto group will convert it into C17 hydroxy group, and this androstenedione will convert it into the new molecule that is known as the testosterone. The enzyme that is involved in this step is the 17-beta hydroxysteroid dehydrogenase. You may observe that this is the structure of androstenedione, and now what is happening? It is being acted upon by the 17-beta hydroxysteroid dehydrogenase, and after that it will undergo through the reduction at this position. Reduction at this position will take place, and this ketonic group will be converted into the hydroxy group, and this is the chemical structure of testosterone. And you may observe that this ketone group is now being reduced and it will get converted into the hydroxy functional group. This makes it the testosterone molecule. So the chemical structure of testosterone has the C17 ring nucleus, and it has the ketonic group at the C3rd position and the hydroxy group at the C17th position. Metabolism of androgens. Metabolism of androgen or testosterone occur like other steroid hormone. So the enzyme that is involved in the metabolism of testosterone or androgen is the 5-alpha reductase. 5-alpha reductase will convert this into the product that is the 5-alpha DHT. In many tissues inside the body, wherever testosterone is being present, will be metabolized. This will occur either to convert the testosterone to the more potent androgen, means more potent androgen, because these DHT that are the metabolite of the androgen or testosterone are the more potent in action. They, like in the case of the prostate. Further, the primary route of the metabolic inactivation of testosterone and DHT means the next step will be the oxidation to the 17-on, means the 17th position hydroxy group will be oxidized and will be converted into the ketone functional group. And the 3-on, the third position ketonic group, will be reduced and will get converted into the 2 intermediate product like 3-alpha alcohol or 3-butol. 3-alpha will be the major byproduct. The androsterone is the major urinary metabolite and was the first androgenic steroid that is being isolated. These metabolites are primarily excreted mainly as the corresponding glucuronide, means in the phase 2 metabolism they will be conjugated with the glucuronic acid and this will form the glucuronide more polar conjugates, and ultimately they will be excreted out in the form of glucuronide. So this is the chemical reaction. In this reaction you may observe that this is the testosterone or androgenic molecule. This is being metabolized. This reaction shows the metabolism reaction of testosterone. It will be acted upon by the 5-alpha reductase enzyme, and ultimately this will cause the reduction at this position and the new 5-alpha dihydrotestosterone is formed, which is abbreviated as the DHT. So in the next step it will be further converted into the other inactive metabolite, like we have observed that 5-alpha ol, that the reduction at this position, and beside this the oxidation at this position, that it will be converted into the ketonic group. Testosterone USP. This is the testosterone formulation that is available for the therapeutic purpose. Testosterone IUPAC name is the 17-beta hydroxy androsten 4-ene 3-on. This is representing what? That it has the hydroxy functional group at the 17th position, androsten that is the nucleus, and the 4-ene represent the double bond at the 4th carbon, and 3-on third position has the ketone group, which we have already discussed. So testosterone that is being naturally available has this chemical name. So in the woman it basically mainly acts as the precursor. Precursor for what? Precursor for the formation or biosynthesis of estradiol or estrogen, and we know that the estrogen is majorly being synthesized inside the female body. So testosterone, which is if formed in the excess in the female, will be get converted into the estrogen. It is rapidly metabolized, means this testosterone which is naturally occurring, or if we formulate the testosterone just like the naturally occurring testosterone molecule, is orally inactive. How inactive? Because it is being rapidly metabolized at the C17 position. However, it has a significant oral activity. Testosterone is available in the transdermal formulation, gel formulation, and the implant formulation, and beside this intramuscular depot formulation are also being available. The purpose of such formulation is because the oral formulation is basically orally inactive, means it is rapidly metabolized. It has the high rate of metabolism. and the drug is being rapidly inactivated. So for the purpose of prolong its action to achieve its therapeutic action which is required for the body, different formulations are being made, like transdermal formulation, intramuscular depot formulation, and such formulation are being formed by the ester formulation. The ester derivative of testosterone are being made at the 17th position carbon, like the different type of USP available testosterone formulation are the testosterone propionate and testosterone 17 beta cyclopentyl propionate and the other derivative. These all are representing the different type of ester of testosterone, and these testosterone are basically orally active because they have the ester group at the C17 position, which will reduces its rate of metabolism. Like this reason we have already discussed in the biosynthetic derivative of estrogen as well. So this naturally occurring testosterone is basically orally inactive due to its rapid metabolism and high first pass metabolism, and that's why this is being converted into different formulation. Testosterone andeconate is the ester, and this, as I have already discussed in the previous slide, that it is the orally inactive formulation because of the presence of ester, which prevents or decreases its rate of metabolism. The synthetic derivative are the like mestrolone. Mestrolone is nothing but it has an additional methyl group at the carbon number 1 position in comparison to testosterone. This mestrolone is also orally active formulation of testosterone. Another formulation is the nandrolone. Nandrolone or nortestosterone. In comparison to the testosterone, nandrolone do not have the methyl group at C19 position, but it has the more anabolic activity than the androgenic activity. Nandrolone is mainly involved in the anabolic functions, like in the increase of the muscle and bone growth, as we have already discussed in the previous slide. The anabolic function of anabolic function of androgen hormone. Now this is the testosterone SAR structure activity relationship of the testosterone. The basic ring nucleus that is present in the testosterone is the carbon 19 ring nucleus, and the name for the carbon 19 nucleus is the androstene nucleus. So you may observe that this is the C17 ring 4 ring of steroid nucleus. This is the carbon 18. This is the carbon 19. This makes it the androstene nucleus, and this androstene nucleus is carbon 19 because it lacks the side chain of the carbon that is present at this C17th position in the pregnenolone. So in case of pregnenolone, we have the additional 2 carbon side chain at this position. So the removal of this 2 carbon side chain at C17 position will make it the androstene or C19 nucleus. Its structure has the ketone group at the 3rd position and carbon-carbon double bond at the 4 and 5. And beside this, the hydroxyl group is present at the 17th position. This you may observe that the testosterone chemical structure has the hydroxyl group at the C17th position. The different type of synthetic derivative are formed from the testosterone to make the more effective oral formulation, like mestrolone is one of the synthetic derivative that is being synthesized by the additional methyl group at this carbon number 1 position. So if testosterone have the additional methyl group at this position, this derivative will be termed as the mestrolone, and it also lacks the double bond at the carbon number 4 and 5. So the mestrolone do not have the carbon-carbon double bond between the 4 and 5 position. There is another synthetic derivative, that is the nandrolone. Nandrolone is that type of derivative that lacks the C19 carbon. Okay, and that nandrolone has mainly the anabolic activity. Different type of ester derivative are being formed by the substitution at this C17 hydroxy group. Means this C17 hydroxy group will be reacted with the different type of acids being substituted at this site, and now ultimately it will get converted into the different ester derivative, and beside this alkynyl derivative will also be formed at this C17 position.